Hello and welcome to Chapter 20 in EFB103. This is all about phylogeny. So understanding phylogeny is all about trying to understand the evolutionary history life on Earth. And so if we want to understand that, we might be interested in relationships of extant living organisms or relationships of fossil specimens. Where do those fit into the history of life on Earth? And to understand this or to visualize this, what we can do is create an estimate of phylogeny. Phylogeny is the actual evolutionary history, and then we create these trees, phylogenetic trees, that are a reflection of this, or our estimate of what phylogeny was. One of the patterns that we typically see in a phylogenetic tree are that organisms that share characteristics tend to be grouped together on that tree. And that makes sense because those are organisms that have a recent or shared evolutionary history. And so to construct these estimates of phylogeny, we might look at morphological or genetic information. And again, the organisms that are closely related are going to typically share many morphological features, more than groups that are less closely related. And at a genetic level as well, they share genetic similarity or share different kinds of genes that you say don't see in other less closely related groups. So all this information is very useful. The discipline of systematics, this is an important one that relates to phylogeny. This is all about classifying organisms to understand these evolutionary relationships. So in systematics, we're trying to put that puzzle together, put those pieces together in the right way to understand how phylogeny happened. A key piece of systematics, a part of that, it's its own field really, but it's very important in systematics, is taxonomy. So taxonomy isn't about the phylogeny per se, but this is about creating ordered subdivisions. So think, kingdom, phylum, class, order, family, genus, species, those groupings, and then naming organisms. And when you think of taxonomy, you might think of Linnaeus. So he's the one who really came up with our modern day taxonomic scheme. And there's a couple of things that are really critical that we can draw from what his work was. This hierarchical classification. So we have these groupings, kingdom and phylum, for example. And notice kingdom is the bigger group here in this particular example I'm pointing out. And within that, the phyla fit within that. So you have this nestedness in this hierarchy. The other important thing that Linnaeus came up with is binomial nomenclature, so that each species is given a particular name that represents its genus and species. An example here of shameless self-promotion here, this is a species that's named after me. This is called Kudoa whipsy. This is a fish parasite. It infects the muscle of this fish, Abdiphtha bengalensis. And it's an example where we see how we write species names. There's a few things. One, you need these two words together that make the species name, the genus and species, or specific epithet. That is the whole name. The first letter of the genus is always capitalized, so there's a capital K here. And then when we would also italicize the species name. And we don't do that with higher taxonomic groups, like say family or order. Those are not italicized, but we do that with the species name. And again, together, that's what makes the entire species name. If you're not able to italicize, say you're writing this on a form for a class or something, you can underline it. It's a way of emphasizing that this here is a species name. All right, so here's that hierarchical classification, just a reminder of that. Again, notice the nested groupings here, and each taxonomic unit, this is an important term, it's called a taxon. So if we have the genus Homo, for example. Homo is a taxon. That's like a name. Now, Homo itself isn't anything. You need to, if you're talking about a species, for example, you need that whole species name. So here's the species, Homo sapiens. That is something, right? These other ones are just categories or taxa. Taxa is the plural of taxon. I will say I noticed in this figure here that these like family, order, these are italicized, and that would not be how we would typically write them. I think this is just done for emphasis here. Of course, again, genus and the species name would be italicized. So when, Linnaeus was putting all these groups together, he focused primarily on morphological characters. Which ones are similar? How should we cluster organisms together based on these similar characteristics? And we still do that. Again, we start to take more molecular information, for example, DNA sequencing. But characters that we might use in one group may not work for another. And a great example would be, say, vertebrates versus invertebrates. Here we have a snail. If we were interested in categorizing snails, we might look at the shape and the direction of the spiral of the shell. That's not going to be a useful characteristic for categorizing, say, felines or mammals in general, any vertebrate animal, right? So that's not going to be useful, and vice versa. And within sort of lower taxa or narrower taxa, maybe that's a better way to think of it, say at the family level, there might be characteristics that are useful across the board for all the organisms in that family. But if we get further and further out to, say, kingdom level, that might not be as useful. A characteristic might not cover all the organisms within that kingdom. And we have to look to lower taxa to find those specific characteristics that are useful within those particular groups. All right, so let's bring back this idea of the phylogenetic trees now, linking it to taxonomy. And this might be a goal that we have, is to connect taxonomy to the phylogenetic trees. Because ideally, we would want a taxonomic scheme or classification scheme that reflects phylogeny, that shows that if something is in the same family, that those organisms have a common ancestor. And we don't always see that. Sometimes things are grouped incorrectly and named incorrectly, and we have to revise that. But here's an example here. So we layer on our taxonomic groupings onto these trees. It's also important to know that just because something is a genus doesn't mean that there's only one species within it. In this very simple example, we see two species within Canis here. Of course, there's a lot of things missing on this tree. The point being is, just because something's a genus doesn't mean, there's 10 species in that. In some groups, there may be 100 species. In other groups, there may be one. So it ranges widely at all these different levels. And so some organisms are much more easy to classify in these ways that fit what we think the phylogenetic history of a particular group might be. Other times it's a little more difficult, and we'll use an example here of the red panda. And so taxonomists might get a species wrong, a placement of a species wrong, because maybe that organism has lost a key feature that's typically found in that group, or maybe due to convergence, an organism maybe just coincidentally looks like a set of organisms that they actually aren't evolutionarily closely related to. And so that can lead to some of this misclassification. Again, ideally, we want our taxonomic schemes to match up with that actual evolutionary history. So the red panda has been classified in a few different genera and a few different mammalian families over the years. Is it a panda? Is it a bear? In the Ursidae? Is it raccoon-like? They've been classified with raccoons, and there have been others that have been proposed. So it's kind of had been, from a taxonomic perspective, a hard one to pin down. So how do we answer this? Really useful source of information is now genetic data. And this is going to allow us to strip away some of those outward phenotypic similarities that might make classification difficult and look at what's happening underneath at a genetic level and see where organisms are related. And so what we find with the red pandas, they actually form a lineage here in this tree out on their own. So here's the red pandas. They're not sister to the raccoons here. They're not bear. They're not a kind of bear. The ursids, these are down here, a different lineage. The red pandas now are classified in their own family to reflect that they are a phylogenetically distinct grouping. All right, so let's look a little bit more at these kinds of trees and how to interpret them. And again, a reminder that a phylogenetic tree is a estimate of phylogeny, and it can be thought of really as a hypothesis of what we think has happened in evolutionary history. The branch points represent common ancestry. So here's where a split, say, fishes and other vertebrate animals here, say a common ancestor occurred here, and then there was a divergence. Where we have groups next to one another and with a most recent common ancestor here, like chimps and humans, these are called sister taxa. So this is a common term that you'll hear, sister taxa. And you'll look a little closer at this. So again, here's humans and chimps, sister taxa. Something that sometimes people might get confused when you're looking at these trees early on is you can see, oh, well, fish and frogs, they're right next to each other here. Why aren't these sister taxa? We go back, follow the branches back. Look, these branches don't connect together. They're not sister taxa. Where do the branches connect? Ultimately back here. So you could say that fishes and all these other vertebrate animals are sister groups. But that's a pretty that's a pretty long way to go back here to find that connection. So usually when we're talking about sister taxa, we're talking about like the chimp human example. And we'll see that a bunch of times as we look at these different trees. Another thing to pay attention to, and it has to do with that idea of recognizing what's a sister taxon and what isn't, is the fact that, if we were saying, well, fish and frogs, they're right next to each other. Yeah, but look, we can rotate this tree. These nodes allow for free rotation. So if we make these rotations as are shown here, we get this tree. Now, the relationships are still the same in this tree. Humans and chimps, sister. Humans plus chimps against lizards. Those are sister groups together. That was the same over here. We've just moved some things around, and this reveals this idea of you glancing at fish and frogs over here. They look next to each other, but they're not, because we could rearrange the tree, and now you might not assume that. And you look, oh wait, frogs and humans are next to each other. No, they're not. You have to go all the way back here to find that common branch point. So really the key is following back from the ends of these tips of these leaves, if you will, to where they join. That's going to be important for recognizing sister groups. Another thing that's important is, well, where do we root these trees? Because if we root trees differently, sometimes we might appreciate a different looking relationship. And again, if we focus on just these sister relationships, we'll avoid that pitfall. And so usually we're trying to find a place to root the tree that is representative of evolutionary history. And we try to find an outgroup or a basal taxon. A basal taxon, like we see here, G, this is a group that is and has ancestral characteristics often, but it's a way to root the tree, and it's at the base of the tree. That's why it's basal. And it allows us to then more correctly interpret the relationships of what's happening within the tree. Now we can see this in action here. We're going to take first this unrooted tree. So we have 4, let's say, species, A, B, C, and D. And if we don't know what the ancestral character is, or which of these is the basal taxon, we can create this unrooted tree. It's like, well, we know C and D are over here, and A and B are over here. All of these, say, are equally diverged from one another. We don't know which is which. That could be a tree. And sometimes trees are displayed this way. Say, we don't know how to root this tree, so we're just going to give it to you unrooted. But the point of this is we can root this tree in all of these different places labeled here, 1, 2, 3, 4, 5, and we get something that looks different, right? So if we root here at 1, where B is sort of the outgroup here, or the basal group, we see C and D here next to each other, and then A is most closely related. But if we root the tree here, C and D, and then B is next to it, right? So this is kind of important that we're rooting the tree in the right place. And you can follow along and see these different rootings and then ultimately these different relationships. So understanding where to root the tree is really important. Or again, if you're in doubt, you can keep it unrooted and say, we don't know, but we know, say, this one and this one are more closely related to each other than this one and this one. So you might see unrooted trees. Usually at this level, you're seeing much more clear ladderized trees like this, where this is called a vertical tree, where we see the branch points splitting across here, a flat line basically. Here's the direct split, and then the branches of these ancestors extending outward and then connecting up to these other lineages. Or we see a diagonal tree, which is just the same representation of this tree above, just displayed in a slightly different way. Now, again, these trees give us a sense of the pattern of what's happening. The length of those branches don't necessarily equate to phenotypic similarities. We can layer on phenotypic information onto these trees, but it doesn't necessarily mean, well, if this branch is this long, then this organism is 10 times, has skin that's 10 times more green than this other one. Nothing like that. We typically can't quantify it in that way. So it doesn't equate to phenotypic similarity. It's just a pattern of descent and really what's called bifurcation, right? Where we have these splitting, and here's another split here. That's called bifurcation. Trees typically don't tell us how old particular taxa are. Although, as we'll see later in this chapter, we can apply to molecular data something called a molecular clock to try to estimate that along with the fossil record. But a lot of times we don't have that information. We just have this pattern of descent or bifurcation. Another important thing here, it shouldn't be assumed that one taxon evolved from the one next to it. So let's say A here. We could think of a human chimp example. Humans, let's say, are A, chimps are B. Humans did not evolve from chimps, right? It's just that we have a common ancestor. So that's really critical to understand, is sometimes we look at extant organisms, things that are alive today, and think, oh, well, X evolved from X. No, the stuff that's around today might have a common ancestor, but one didn't evolve from the other, right? And again, think about that, back to that example of a family, right? You didn't evolve from your cousins, right? Your cousins aren't your descendants, but your, say, grandparents are your common ancestor. You've got to go back in time on the tree here to find those connections. All right, so let's apply some of what we're learning here. Is when we create these phylogenies, we can reveal information that maybe we didn't appreciate before, or we can start to learn about things we didn't know about an organism if we can put it into a tree and say, oh, this is actually more closely related to this group of species than these guys over here. And that helps us then understand maybe something about the biology of that organism that we didn't previously know. Here, example of shrews and these elephant shrews. Elephant shrews are not directly related to typical shrews here. It's a completely different lineage. These phylogenetic trees can be used to solve mysteries, like this example of where, in your textbook, where different samples of whale meat came from, and or maybe being sold as, or said, this isn't a humpback whale, this is something else. Well, when we take mitochondrial DNA information, plug it into our phylogenetic tree here, we see that this unknown sample here actually is very likely is humpback whale. And so this could be useful information from that perspective, once we have this framework of our tree here. So speaking of genetic information, this has become much more common and very useful. We can use both together, morphological and genetic information together, but we get a ton of information from genetic data nowadays. And when we're looking at either of these, it's important that we're only using homologous characters. So we understand homology from that perspective of, bone structure in the arms, but that's the same when we're looking at genetic information. We want to use homologous genes, not maybe a gene that, well, this kind of looks like it's similar. It might not be, and therefore it might have a different evolutionary history, right? And so that wouldn't be helpful for creating an estimate of what our evolutionary history is. Just generally, the more similar we have, or the more similar a genetic stretch of genetic information is, or the more numerous the homologies are between morphological characteristics, the more likely organisms are to be closely related. And we can take an example here of heart structure in the context of heart evolution. And if we look at, say, mammalian hearts, it's a four-chambered heart. But birds also have a four-chambered heart, right? So if we were just focusing on that character, four-chambered or not, we might lump birds with mammals here. But we see birds actually form a distinct lineage here with these lizards that have three-chambered hearts. And so that's important, why we need to take that total information. So this four-chambered heart has evolved more than once in the history of life on Earth. Okay, so here's a question for you. What is a possible or common mistake that taxonomists might make when attempting to place seemingly closely related species into a shared group? All right, yeah, so the potential pitfall here is if two different organisms maybe look the same, they have similar morphological features, but those features are not homologous, that you might misclassify. And we've talked about this before. This is the idea of analogy. And so homology are those characteristics that have the shared ancestry. That's what we need to construct phylogenies. We need to make sure we're working with homologous characters. Analogous characters really throw a wrench in our ability to create those trees that reflect an evolutionary history. And as we talked about this before, this is called convergent evolution, when you see similar features in different organisms that don't have that shared evolutionary history. And when we're dealing with potential for analogy, another term for that is also homoplazy. It's sort of given the plasticity really of a certain character, they might look similar or converge. So that's where that term comes from. And so ideally, we're working with as many characters as possible, because if we are, we're not going to fall into this trap here. Say, if we're just looking at, organisms that have wings, then we might mistakenly place this pteranodon and birds together in a shared lineage here with a common ancestor. That would be a mistake. So we wouldn't just necessarily rely on wings alone, as we see wings have evolved more than once. And again, that's a homoplazy. That's a character that's evolved more than once. And so if we were to look at, say, bone structure or other characteristics, now we would probably end up coming up with the correct phylogenetic tree, because we're looking at more and more characters. So that's really important, to look at a lot of characters. That helps wash away some of these analogous characters. Now we can have analogy crop up in genetic sequences too. And this is where, again, that character number, looking at as many characters as possible, is really important. And so let's take an example here. We've got two DNA sequences from, say, different organisms. If we just look at something that's 4 nucleotides long, it's not very long or very useful, as we'll see. We could ask the question, what are the odds, just by total chance, that these two sequences of 4 bases are going to be identical. Just by total chance, it's 1 in 256, which is pretty likely. So this could come up, this just by totally random chance. That could be analogy or homoplasy. So it's crucial that we look at much, much longer sequences. So let's say this sequence, which is way shorter than what we would typically use to create a phylogeny, but just this sequence, which is much longer than the 4 base one, the chances that these are going to be similar or identical by chance gets really, really remote, right? So the more nucleotides, the better. Typically, we're using hundreds or thousands of nucleotides as our information for constructing these phylogenies. So let's look at this, how we might approach this. Here we've got 2 species, species 1 and 2, and here is a bit of DNA sequence from each one of these. Now, we're going to look at this from the perspective of the change over time that can occur. These are homologous genes, and substitutions might occur in these genes, insertions and deletions, and just eyeballing this, so these are identical, we could say, oh, well, we can line these up pretty easily, but it gets a little more tricky when a substitution happens or an insertion, and maybe there's multiple insertion and deletions. Which ones are more important? How do we align these? And we use computer programs to do that nowadays. And again, these are potentially hundreds or thousands of bases long. And so let's take this scenario of these ancestral sequences being homologous and the same here. And then over time, this species 1 has a deletion that occurs here. Species 2 ends up having this insertion. Now, if we just try to line these up side by side, we see they don't really match up. This is image 3 here. But we could probably do this by eye, but a computer program would do this. The computer program will insert a gap here and here, and now we can see the rest of the sequence actually does line up. So this is really useful using computer programs to create what are called DNA sequence alignments, that now we can analyze homologous characters, because this G here is homologous to this G here, this T to this T, etc. And potentially there's a substitution here. Let's say this C is actually a T in species 2. That's still a homologous site, but that's actually really informative if we see that a mutation occurred in species 2 here. Now, where it potentially gets a little more difficult is if we have really distantly related organisms, and depending on the gene we're looking at as well, whether that's a slowly evolving or more quickly evolving gene. If we look at these 2 DNA sequences here, 1 and 2, we can align them. The matching bases here are highlighted in yellow. But now we're getting into this realm of, could we have this occur just by chance? And the further and further apart these sequences get, the more likely we might be to mistakenly say that these are homologous genes. Again, depending on the gene and how many alignment sites we have. And so when we're looking at these comparisons, it looks like we have homology, but these are not, these are analogous sites. So we cannot really construct a tree very accurately here. And something that can happen even when we have homologous genes, but there have been so many changes in those genes of distantly related organisms, we can have something happen called long branch attraction. So here's the example. Here's what the true tree, quote unquote, should look like. B and C are sister, and then B plus C are sister to A. D is the ancestral group. But because A and D are so different in our alignment, that just by chance, we might get a slightly better match between A and D. And so that's the inferred tree here. And this, again, it's called long branch attraction. These branches are really long because there are a lot of nucleotide differences. And then just by happenstance, A and D stick together here. And there's ways of dealing with that, but it's important to recognize that's a potential pitfall. All right, so we're building phylogenetic trees called cladograms. We're going to take evidence of from molecular systematics. What are we basing these similarities in? All right, yeah, we need homologous genes. That's what we're after. But if they're identical, that's not very useful. There need to be differences, and those differences accumulate through mutations. And so one of the approaches to constructing these phylogenetic trees is called cladistics. And this is really taking our homologous characters we've identified, whether that's genetic or morphological, and then we're thinking about this from a perspective of common descent and where those characteristics arise within the tree. And so a clade, or this lineage, if you will, it's a group of species that includes all the ancestral species and its descendants. And so we'll see where we get some deviations from that and where our taxonomy doesn't maybe match up with our estimates of phylogeny. We do see this nesting type effect here. So say mammals and reptiles are nested sort of within this broader group that includes amphibians, mammals, and reptiles that have 4 limbs. And then these 3 are nested within this other bigger group, animals that have backbones. And so we see this nesting, which again, consistent with our taxonomic schemes and our estimates of phylogeny. So when we're looking at these phylogenetic trees, we're trying to identify groups that are monophyletic. Mono means one. And so we're looking, say, at this group here, and there's this common ancestor. So A, B, and C are extant species. Those are what are around today. Here's common ancestor number 1, and all of our species group together very nicely here. So this would be considered a monophyletic taxon. And this is a reflection in our classification as well. So everything looks pretty good. We're always striving to identify monophyletic taxa. We don't always find that, of course. So here's another possibility, a paraphyletic taxon. So this is group 2 here. So let's say we've taxonomically categorized species D, E, and F together. Let's say they're in the same family, and G is in a different family. But the problem here is that G shares an ancestor with D, E, and F, but it's not included in the group here. And so a paraphyletic group is a group that contains the ancestor here at 2, this node at 2, but not all of the descendants. See, G is left out. So this is a paraphyletic group. Another kind of grouping that we see, which maybe will look a little similar, but the key here is where the ancestor is a polyphyletic group. So here we've classified, let's say, A, B, C, and D. Let's say we lump them in the same family. But if we look at our estimate of phylogeny, look at D is way over here. It actually doesn't share a common ancestor until we go way back here with A, B, and C. So this is a group that contains species sort of out on the tips of the branches here, but does not contain this ancestor. Here are some specific examples of poly and paraphyletic groups. So here, this is a paraphyletic group here, the hippopotamuses and other even-toed ungulates. To make this monophyletic, we really would need to include the cetaceans within this, and that would, gray out this whole lineage here, including this ancestor. A polyphyletic group example here would be seals and cetaceans. So if we lump these guys together in the same taxonomic group, hey, they're these marine mammals. That's not a grouping that reflects evolutionary history. These guys represent different branches on the tree of mammalian diversity here. All right, so we're really striving to identify what are the shared ancestral characters. That really helps us to create these trees. And they have to be homologous. That's really crucial as well. So those are useful for understanding ancestry and where those deeper nodes connect. But also we're interested in derived characters that seem to appear maybe either more recently or further along in the tree that are in a narrower group. And so we see examples of that here. So hair we see as this shared derived character in mammals. Here we've got primates and rodents, rabbits as examples in this tree here. And an ancestral character, a shared ancestral character here would be vertebrae. This includes all the organisms here. Now, a lot of this does depend on perspective. So a character could be both shared and ancestral. So if we were just interested in looking at mammals, for example, we cut out all the rest of this tree, we're only focusing on mammals. Hare here would be an ancestral character that we find in mammals. Or let's see, four limbs, for example. That's a derived character in amphibians plus all these guys. But if we were only interested in amphibians plus these primates, rabbits, crocodiles, dinosaurs, and birds, that would be considered an ancestral character. So it's all a bit of a matter of perspective. A derived character may not always just be an addition of something, but it might be the absence of something. So here, limbs in reptiles, for example, we see the evolution of no limbs in two different places. And these are derived characters. Now, between glass lizards and snakes, this is not a shared derived character. You'd actually have to think about this as loss of legs in glass lizards and loss of legs in snakes. Those are two different derived characters. Another set of terminology to be aware of is this idea of in-group and out-group. And we've talked a little bit about this when we were looking at rooting phylogenetic trees. We're trying to root with an outgroup, and those taxa at the base of the tree are the basal taxa. Those might make for good outgroups. And so we're trying to identify, based on other knowledge, well, what would be a good outgroup? And sometimes, again, maybe we don't know that initially. We have to do some other kind of analysis to figure that out. But applying our derived and ancestral characters to this model, if we find the same character in our outgroup and ingroup, those are typically thought to be ancestral. Again, this is relative, right? It depends on how far we're going down in the tree. And then those characters within our ingroup, or rows within our ingroup, those are our derived characters. It is, of course, very useful in figuring out where and when a character might have arisen within our tree, because when we get past that point, let's pick four limbs here again, then anything that falls within that, we would expect to have four limbs. And if they don't, that might represent a loss of that character. And so we can reliably say, okay, anything with four limbs is going to be in here somewhere. Frogs, turtles, leopards, and whatever else fits in here. A lot of stuff fits in here. These are just representatives. And then we can learn something about the biology. We can understand something about the relationships. And so understanding where those first appear, where should we mark our tree with that characteristic appearing, can be very helpful. Now, we mentioned earlier how branch lengths might not be relevant for understanding, say, time, which we'll see an exception to that. We talk about molecular clocks here. But in some trees, branch length might represent, say, genetic distance or differences between shared characteristics. The branch lengths might represent that. It might not just be a bifurcation. And so in this example here, we see branch lengths of different lengths. We're not just seeing even length branches with splits. As we see here, the branch lengths are all different. And the length of these are a reflection of the genetic differences between these organisms. We can, as I mentioned, we can potentially apply a clock to this. And we would calibrate this based maybe on the fossil record. And we can then overlay our genetic differences with time. So we can say, well, we know, say, humans and mice share a common ancestor back here 70 million years ago or so. And that's where we can split it. And then we can look at those numbers of genetic differences and determine, well, how many mutations are occurring every million years, for example. And we'll come back to this concept when we talk about using molecular clocks. Another really important thing when we're building these trees is deciding what the best tree is. because potentially, given especially if you have a lot of different organisms, there might be many different ways you can put that together. How do we determine what's the best tree? And there are many approaches to this, but one approach that we'll talk about here is called maximum parsimony. And so if something's parsimonious, it's sort of cheap, if you will. It's basically a way to find the tree that is a result of the fewest steps, the fewest changes in evolutionary history. That might be a character or phenotypic character change, or that could be genetic changes. So we're trying to find the tree that minimizes those changes. And so our example trees down here, we have 3 possible trees being produced from these data from taxon A, B, C, and D. And it's this first tree that you only need 2 changes occurring here. They're marked on the tree here, yellow to blue and square to circle here over in this lineage. That only requires 2 changes. So this one on the left would be considered the most parsimonious tree. Now, it doesn't mean that these other ones aren't possible. It's just that, given the weight of evidence, what is most likely? Probably the one that requires the fewest changes. And so identifying the minimal amount of changes that get you to that potential phylogeny is key. We use computer programs for this, because even if we were looking at just 4 taxa, here are 15 possible trees that could be produced. So if we're just looking at it cold and we said, we don't know what the tree would look like, maybe we don't have any other data, there could be all these different trees, and then we would crank through a computer program which would identify, well, which of these trees is most likely at fewest steps. How do we minimize those changes of, say, nucleotide data or other character data? Which of those trees gives us the best, or which of these trees is sort of the lowest scoring, aka the most likely given the data? And we can walk through an example here with these beetles here. We just have 3. And so there are 3 possible trees that could be produced here. 1 and 2 are sister, and then the third is the basal lineage, or 1 and 3 together, and 2 is the basal lineage, or 2 and 3, and then 1 is the basal lineage. And so we could just say, all right, here are totally blank. We don't know. It could be any one of these. Let's look at our genetic data here. Again, this is pretty small compared to what you'd actually look at, but it helps for example. And we can look at this first site. So site 1 here, we've got C, A, and A in our different beetles and ancestral sequence. And so if we apply this model to any one of our trees, how do we get this change? Well, if our ancestral was A, all we need to do is change an A to a C here, and that would give us 1 and 2. Those guys have a C, so that would work. If we take our other tree, this is one of our other trees, 1 and 3 are sister, to make this site work here, you'd have to change a C from an A here and here. So there's 2 steps in this tree. And then likewise in this third possible tree, there's 2 steps. So we can do the same. We can apply that same logic now to these other sites. So here's site 2, actually going to be pretty much the same as site 1 if you see how the nucleotides stack up here. 3 is going to be a little different. So we see where 3, we need a change here and here in our first tree. But it's cheaper over here in tree number 2, only need this 1 change. So you start to see it's not always black and white. Not all the sites are going to be the same, but we add them up. And what we end up with here is our first tree. Given our data set, we can get this outcome with only 6 nucleotide changes, whereas both these others require 7. So this first tree is really the most parsimonious, and therefore we'd say it's the most likely. All right, so thinking about parsimony, according to the principle of maximum parsimony, what can we say? Right, so it's the tree that has the minimal change, or the fewest steps, that's the most likely. That's the most parsimonious tree. All right, another thing that we have to remember is phylogenetic trees are hypotheses. These are estimates of phylogeny. So these are our estimates of what we think actually happened. That's the phylogeny, and the tree is a representation of what we think happened. And just like any scientific hypothesis, as we learn new information, we might modify that. So a great example, and we've already heard about this already, is we might have a tree that's based on morphological characters. That's all we had until the more modern era. And when we apply, say, DNA evidence, we might move some things around. So let's look at the relationship of whales here. Here's whales, sister to the mesonychids here. And so this is before DNA evidence. You see some other relationships here too. Horses are the basal group, for example. But after we apply genetic data, we see that hippos and whales are sister. Camels are not sister to the ruminants like we saw before, but these are different lineage here. The mesonychids, those are the basal lineage before the horses here. So we change, we learn new things, and we modify. That's how science works. And we get more information, we build that up, and that's, the foundation of, scientific theory. All right, so from these hypotheses, we can start to piece together information about other organisms that fit into our tree, but maybe we don't have living specimens of, for example. So we know something about birds, we know something about crocodilians. These are extant species, those are around. But these other related organisms, so dinosaurs, different kinds of dinosaurs here, these have gone extinct. But based on what we know about birds and crocodilians and the overlap that we see, dinosaurs are phylogenetically bracketed. This is a term that we use, that they're in between. And so we would think that features that might be common between crocodiles and birds, those might also occur in dinosaurs as well. And you could apply this to any group, but we're using the bird dinosaur example here. And so, based on this kind of information, so we can look at, here's what birds and crocodiles share: four-chambered hearts, singing, nest building, brooding behavior, is that now we can say, well, we'd expect to see some of those things in dinosaurs. A lot of these characteristics would be pretty difficult or impossible to find, but we know dinosaurs built nests based on fossil evidence. Based on this particular example, we see fossil evidence of brooding behavior, which we see in birds and crocodiles. And so this is really cool. By learning about the groups that are around or closely related phylogenetically to some unknown group, we can learn more about what might be in that unknown group. Those are hypotheses to be tested, of course. And if we can find evidence, like the case here with this dinosaur brooding, like a bird or crocodile, then that supports that idea, and we can then build on that. We learn something else new, and that helps support our tree. Or maybe we don't find evidence, or we find evidence to the contrary, and that makes us rethink our tree. All right, so something I've mentioned a couple of times so far is this idea of using a molecular clock. And the question that we're trying to get at here is, when we make those trees, can we learn something about where those branch points might have occurred in time, and therefore how long certain groups have been around, or where we might predict to find ancestral groups, and so on. So we can ask all kinds of questions if we can apply a clock to these trees. This molecular clock is based on genetic information. So we're looking at rate of mutation over time. And it relies on this idea that mutations in a particular gene are going to, say, occur at random, and the rate is relatively constant. We'll see that that's not always true. There's things that might influence that. And some genes, the rate of mutation is different than in other genes. So what we're looking at in this figure, cytochrome C here, has a much lower rate of mutation than, say, hemoglobin. We see over time more mutations in hemoglobin. We'll talk about why that might be in a little bit. But if we know that rate, then we can look at the genetic sequences between organisms, look at the number of differences, count them up, and say, well, if there's 30 differences there, that equates to 10 million years, or whatever our clock says. Our clock sets that time. So how do we calibrate that? We really need to have fossil data. So if we know that, okay, we have this living organism and this related living organism. Where do we see in the fossil record where they likely diverged, or where do we start to see, I don't know, let's pick something around modern day horses, or some ancestor of a horse and their living relatives. Where do we find that? And that helps us calibrate the molecular clock. So if we say, okay, well, there's 100 nucleotide substitutions in a hemoglobin gene between these two organisms, and we know based on our fossil data that their common ancestor was around 30 million years ago, or whatever it happens to be, that allows us to determine what that rate is of change and as it applies to the number of years. Now, there are some challenges to this. Genes don't necessarily evolve in a very regular way. Some regions of a gene might evolve in irregular bursts. I've already mentioned some genes incorporate mutations at a greater rate than others. Here's an example of a virus. Usually we're not using viruses to construct these like deep phylogenies, but for shorter times, we might look at a virus. And I like this figure here. It shows a rate of mutation of these different virus lineages. These are sister groups, but we see, based on the data here, because we have data in real time in these, this short window of years, we can figure out what the mutation rate is, and it varies. And so this is probably happening as well at, say, mammals and fossils in the last, tens of millions of years. And so why might we see differences in clock speed? Well, some mutations are selectively neutral, and that would be like a silent mutation here. We've talked about these before. So a mutation occurs in this codon, in this gene. This thing did code for a lysine, still codes for a lysine. So this is not a mutation that will be selected against. It's called a neutral mutation. And those can accumulate at whatever the base mutation rate is. Now, these other kinds of mutations, like a stop codon or maybe a missense, stop codon nonsense mutations probably going to be selected against. So if those occur at random, that organism, that population, that might be very negatively impacted. Similar with a missense, maybe some amino acid substitutions could be okay. It looks sort of behaves similarly, that almost neutral, or it could be a completely different amino acid that has totally changes the structure and function of that protein. Those are going to be less tolerated. And so it really boils down to how important is that amino acid in that particular sequence, or just going back to the DNA level, how important is that either codon or region of DNA for maybe just producing a piece of RNA, like transfer RNA or ribosomal RNA. And as we've seen this figure already, this is just a different look at it, that rate of mutation is different. In something like cytochrome C, it's fairly low compared to hemoglobin. Now, if we think, where is cytochrome C important? That's crucial in the mitochondrion. And think cellular respiration, that electron transport chain, cytochrome C, is really critical there. So probably there's mutations that could occur that would be very bad, and those are selected against. So if those happen, that individual maybe the mitochondria don't function at all, or has a lower function, that's going to impact the fitness, and we'll see less of that occurring. So that'll appear to slow down the mutation rate. And so the clock rate will depend on the gene that we're looking at. All right, so which thing would, if acting upon a gene, prevent the gene from acting as a reliable molecular clock? All right, so the answer is natural selection. So if selection, say a beneficial mutation occurs, that's going to take off, right? Become more common. So it's going to seem like that mutation is more prevalent or occurred a longer time ago if we think of it from a molecular clock perspective. Or if a mutation is very negative, has a negative impact, it's maybe going to disappear. We never actually see it. So there's mutations that are occurring, a mutation rate that's happening that we're not appreciating because we only see the mutations that can be tolerated, right? 10 million years later. So natural selection is really critical. And this is probably one of the more important ones we have to watch out for when coming up with our molecular clock here. So we could look, and this is an example here, across a genome of a protozoan species, looking at, say, different chromosomes. That's what's numbered here. And we can look at different regions and look at the mutation rate in different regions of a chromosome. And what we see, it's not even. You'd expect if mutations are random, you'd expect a flat line. There'd be the same average amount of mutations at any particular place on a chromosome. These bars would all be the same height, but they're not. Some areas we see a pretty tall bar here, like in red. That's a higher number of mutations versus blue here, these low bars. And the low bars are probably areas that don't tolerate mutation well. Maybe it's a really important amino acid, part of an active site of an enzyme, for example. And so that cannot tolerate a mutation, whereas these tall red bars is places, maybe it's that third position in a codon. It doesn't matter if that's a C, G, T, or A. No big deal, and those mutations can be tolerated. So that's going to definitely influence that molecular clock rate. So we know about some of these pitfalls, but it's still very useful, and especially when we can calibrate our genes to the fossil record. It's when we get to organisms maybe we don't have good fossils for, where it becomes hard to calibrate, and we have to make some assumptions and start to fill in data maybe in a different way. But still, the idea of a molecular clock is still very fascinating and very useful. Here's an application in a smaller window of time and focusing on a virus, which viral genomes typically are fairly fast evolving, quote unquote, because they incorporate mutations more quickly due to their usually sloppy polymerases that copy their genomes, and rapid replication times and so forth. And so we actually have a ton of data on HIV and genome sequences back into the late 1980s here. So these little black dots here are genomes from these different years and the index of base changes in gene sequence. And if we look, we can see depending on how we look at this, a downward trend looking back in time or upward trend looking forward in time of base changes. And we can project backward through time. And we're getting back, say, into the 1960s and 40s when we don't have genetic samples to test, but we can estimate based on a molecular clock that HIV emerged somewhere probably between the 1930s and the 1910s, depending on what kind of model we run. So this can be very interesting from in this case, a disease standpoint, not just sort of a deep evolutionary history standpoint. Speaking of deep evolutionary history, we can also think about how to apply phylogenies from the perspective of understanding all of life on Earth. And one of the big changes in semi-recent years has been this change from thinking of 5 kingdoms of life. That for a long time, organisms were grouped into these 5 kingdoms. What we know now based on genetic data is we split life into these 3 domains. We've talked about these before earlier in the course, the eukaryotes, the archaea, and the bacteria. And there's a few things to note here is, well, one, the prokaryotes, archaea and bacteria, are not sister group here, although, some phylogenies on different genes actually might show that relationship. And what we used to think of, and so those were the monerans, the prokaryotes, the protists, plantae, fungi, animalia, those guys are all within this domain eukarya over here. All right, so here's a question for you. We based on modern data, we have a 3-domain system. What kind of thing could we call this scientifically? All right. Yeah, this is a theory because it's based on a lot of data and evidence accumulated over time and been tested from all kinds of angles. And this is where we've arrived, is this 3-domain system of life. So this is a scientific theory. All right. So this I already pointed out that what we had were these groups: plantae, fungi, animalia, protists. These are incorporated now into the eukaryotes. Notice that the plants, fungi, and animals, these are multicellular organisms, and they're not monophyletic. Plants are a separate lineage from fungi, so multicellularity has evolved more than once in the history of life on Earth. The monarans, as I mentioned before, are not monophyletic because you've got the archaea and bacteria as distinct lineages here. And, similar to the protists, they crop up at all places in this eukaryote tree. All right, the last bit of information to talk about in this chapter is this interesting idea of the role of horizontal gene transfer, or HGT. Here's, ideally this tree of life where we've got our 3 different domains. And a lot of the early trees, and still today, we use ribosomal RNA. Remember, every living thing has ribosomes. So if we want to compare all these things, let's use ribosomal RNA, because every living thing should have these genes, right? There are other genes as well that we can use, and fairly conservative genes like the ribosomal RNA that give us similar results. Here we see eukaryotes and archaea close together. And some trees will give us bacteria and archaea close together. So it might depend on what gene you're using. And because this is such deep time, it can be difficult to pin down those branch points. But the thing that we see throughout the history of life, and why we might get some of those conflicting trees, is because genes have moved laterally between species through horizontal gene transfer, not through like homology and bifurcation of these branches, but a gene jumps from one organism to another. It's sort of similar to how we talked about analogy. If we're using a gene that's jumped from one thing to another, it's not going to, that tree isn't going to represent that phylogenetic history of that gene because there was that jump. And so there's many ways that this can happen in all sorts of organisms. Prokaryotes exchange this kind of information very regularly. It can happen in eukaryotes too, less commonly. Here's an example of what's something called a transposon that can jump from one chromosome to another, or again, between organisms. And we see this kind of horizontal transfer. If we look at the tree of life, this was probably very common in early life on Earth. Still occurs a lot in prokaryotes. Where do we see horizontal gene transfer in eukaryotes? More recently, again, we're talking millions of years, but relatively recently, that acquisition of chloroplasts or mitochondria. So we see those genomes associated with eukaryotes. Again, some transposons will jump genes around, but we still see these lines crossing here in prokaryotes. And we really can think back, the tree of life, it didn't necessarily start at a single point, but you sort of almost have this bush of living things where the branches are crossing over, or as the textbook describes, more of a tangled web. So this is really much more interesting probably than we can really represent in a simple bifurcating tree. All right, that's everything you need to know for this class about phylogeny.